Okay, welcome to the next section. We are going to speak about, we're going to have some great talks about decision making from reinforcement learning, expectations, lapses, and I think without further ado, we're going to start with our first speaker, Bruno Averbeck from NIMH, and he's going to talk about neural systems underlying reinforcement learning. Please join me in welcoming Bruno.
All right, it's working. Okay, good. First, I'd like to thank the organizers for inviting me. I've been coming to this meeting a long time. Sarah said that she has never been here before, and this week I actually looked, and I think the first time I came to this meeting was in 2003. So it has grown a lot since then, and in fact, that meeting had a different name. I think I met Christian at that meeting, actually. We used to talk about, neural coding and that kind of thing. So this meeting has been very important to me as I've developed as a scientist. All right, so I'm going to be talking about neural systems underlying reinforcement learning. And just a little bit of background on reinforcement learning. Many of you here probably work on reinforcement learning. So when you work on reinforcement learning in artificial agents, you're trying to train an agent to select actions based upon states of the world. And the current state of the art, of course, is to train agents to select actions in very complex environments. So there are many different possible states or configurations of this board, and there's many choices you can make at each point in time. And the goal of these agents is to select actions that guarantee some long-term average expected reward over some sort of time horizon, okay? But the current state of the art is the interest in these very complex state action spaces. And the environments, however, are stationary, right? So the rules of the game here are fixed. The game is complicated, but the rules are fixed. When we study these same problems in biological systems, this is where I work mostly, we tend to work in fairly simple state and action spaces. So we like these bandit problems, and we call these bandit problems because they're equivalent to walking into a casino and trying to figure out which of these slot machines to play. And of course, a slot machine is a one-arm bandit because it steals your money. So we call these bandit problems. This would be a three-arm bandit problem. So we often study these bandit problems, but we also often study these in non-stationary environments. So we'll have drifting bandits, for example, or we will have the bandits change. So one bandit that had been good becomes the bad bandit, and the bad bandit becomes the good bandit, because we think this ability to track changes in the environment is kind of a hallmark of intelligence. This is also a little bit historical, but we tend to focus on these sorts of problems. So the neural circuitry that underlies reinforcement learning gets a lot of attention. There's a lot of attention on dopamine and the dopamine projection to the striatum. So this is a coronal section through the anterior striatum in the monkey. Dopamine codes reward prediction errors, which are the differences between the reward received at some point in time and your estimate of what you expected. That's the value of the current choice that you made. And those reward prediction errors are fed into the striatum. And it's often thought that the striatum maintains value estimates, and it integrates these reward prediction errors to drive changes in value, okay? And this has been arguably one of the most successful sort of theoretical models in systems neuroscience. And when I started a while back working on reinforcement learning, I certainly started working on it from this point of view. But of course, the striatum, and all dopamine as well, is embedded in a much more complex set of circuitry. And there's evidence from a lot of different labs that actually all the different nodes in this circuit play important roles in this learning process. I think we don't know that much yet about the relative contributions of each of the nodes in this circuitry to reinforcement learning. That's a lot of what we work on. But this circuit here, I often call the ventral circuit because this connects ventral parts of prefrontal cortex. So this is orbital frontal cortex, ventral medial prefrontal cortex, to the ventral striatum. And then back through these return loops in the thalamus back to cortex. This circuit we think is very important for associating stimuli. In the primate, we usually study visual stimuli to rewards. And we think that the amygdala plays a very important role specifically in this circuitry, and I'll show you some evidence consistent with that in a couple slides. There's another circuit through the striatum, so this I call the dorsal circuit because this connects dorsal parts of cortex into the dorsal striatum. This is the return loop. So these are the cortical, basal ganglia, thalamocortical loops here. And we think this blue circuit is important for spatial cognitive processes. So I tend to study it from the point of view of learning eye movements that return rewards. And I've kind of shown this here as two separate circuits, so a blue circuit and a red circuit, because that's how we think about them in the lab and often study them. But there's actually a continuum of circuitry connecting all parts of prefrontal cortex into different parts of the striatum. And the learning in each circuit probably has mostly to do with the kinds of information brought into that part of the circuit from cortex, okay? So in the first couple of studies that I'm going to show you, I'm actually going to focus on the subcortical circuitry here. And my main goal is going to be to show you that the amygdala plays a fairly important role in these reinforcement learning processes, and then maybe to specify the role of the ventral striatum in a little bit more detail. So this is an experiment done by my previous postdoc, Vincent Costa. He's off starting his own lab at the Oregon Health Sciences University now. So in the first experiment, we're going to compare behavior between three groups of animals. We have a group of normal control animals, and then we have a group of animals with ventral striatal lesions, and a separate group of animals with amygdala lesions. All the lesions are bilateral, and the ventral striatum is both the core and the shell, if you work in rodents, and the amygdala lesion is both the basolateral amygdala and the central amygdala. So this is the task that we used. This is kind of a standard primate task. The monkey's seated in a chair. He's looking at a screen. We're monitoring his eye movements, and he's using his eye movements to indicate his decisions. So he starts out by fixating a central fixation point. Then we present two images left and right of fixation. He makes an eye movement to pick one of those images, and he's stochastically rewarded. So the way we structure the task is that it's carried out in 80 trial blocks. And at the beginning of each block of 80 trials, we introduce two new images that the monkey's never seen before. We assign to one of those images a high reward probability and to the other image a low reward probability. We use these different schedules, and the schedule remains fixed for a block of trials, okay? So if it's an 80-20 block of trials, and this is the high reward probability stimulus, when he picks that stimulus, he's rewarded 80% of the time, and when he picks that stimulus, he's rewarded 20% of the time. And I apologize for the other half of the room, but I have to sort of think through my pointer here. So at the beginning of the block, we introduce these two new images, and the monkey has to sort out what the good image is by trial and error. So in each trial, he picks one of the images, and he's rewarded or not. He learns what the good image is, picks that as many times as possible. Then on a randomly chosen trial between 30 and 50, we switch the stimulus outcome mapping, so the good image becomes the bad image, and the bad image becomes the good image. I'm not sure if I mentioned it, but these images are randomly showing up left and right of fixation. When the stimulus outcome mapping switches, he has to detect that switch to the other stimulus, and finish the block. At the end of the block, we introduce two new images, does another block of 80 trials. At the end of that block, we introduce two new images, and he does this over and over and over again. We get about 2,000 trials out of the monkey per day, so we get 20 to 25 blocks of trials a day, and we just have him doing this learning over and over and over again. So this is the effects of the lesions on learning. So what I'm plotting here on the y-axis is the fraction of times the monkey picks the best initial cue. The x-axis is my 80 trial block. So of course, many, many trials average together, and several monkeys also average together. This is the 80-20 condition. We have the control animals in black, and we see that the animals quickly learn the best image, and they pick it over 90-95% of the time. And then when we reverse, the dotted line here is reversal, the stimulus reward mapping. They reverse their preferences, start picking the other object, and finish the block. We're smoothing in time here. The animals don't actually switch before in the 80-20 condition. In the 80-20 condition, they switch about 2 trials after. But basically, the animals perform really, really well in this task. They learn very rapidly, particularly in the easy conditions, and they do well. So the blue line is the animals with ventral striatum lesions. They have a substantial deficit relative to the control animals. And the red line is the animals with amygdala lesions. They have an even larger deficit, okay? So in general, this is thought to be a very striatal-dependent task, but we see that with amygdala lesions, we actually get, if anything, an even bigger deficit than we got with the striatal lesions. So that's the 80-20 condition, that's the 70-30 condition, and this is the 60-40 condition. Remember, these are run in separate blocks of trials. Across conditions, the learning gets worse in all 3 groups. Here they really struggle to learn, but the same rank ordering of performance is maintained across these 3 groups. I actually used the different schedules because there was some discrepancy in some of the older literature about different behavioral effects, and I thought some of it might be schedule dependent, but we've never really seen a schedule dependent effect all the effects tend to just scale across these schedules. So we see a pretty substantial effect in the animals with amygdala lesions. And there was an older literature, actually, where I came from that suggested that animals with amygdala lesions did not have deficits in what were called reversal learning tasks. And these were old monkey behavioral learning experiments where you had a Wisconsin general testing apparatus and the monkeys were sampling objects. And there are many, many differences between those experiments and the experiments we run. However, one of the differences that seemed to be most salient to us was the fact that everything we ran was stochastic rewards, and the old experiments were all deterministic rewards. So you got a reward if you picked up the object, it had a peanut under it, and in every trial that object would have a peanut under it for a while. So we ran a deterministic version of the task, and when we ran the deterministic version, this came after all these experiments. All the groups improved. The control animals learn really after a while in basically one trial or two trials what the good object is. All the groups improve, but we still see this same rank order of performance. And I'll point out that the amygdala lesion animals, in 30 or 40 trials, they're still only picking the good option about 80% of the time, and when we reverse, it takes them quite a while to reverse. So just to summarize study 1, the lesions show that the amygdala and the ventral striatum both play a role in this kind of stimulus outcome learning. And it was kind of implicit in the previous slide, but our hypothesis about why you would bother having two structures, both doing the same kind of learning in parallel, is that the amygdala is learning quickly and the striatum is learning slowly. So each structure is sort of optimal for different kinds of environments, and they can learn in parallel. And what I mean by that is the following. So when we lesion the amygdala, assuming that only the amygdala and the ventral striatum for the moment are doing the learning, I think there probably is learning in other structures as well, but assuming it's just the amygdala and ventral striatum, if we lesion the amygdala, we're left with the striatum, right? So the red line is really the striatum's ability to learn in these situations, and the blue line is the amygdala's ability to learn, right? So the amygdala lesion animals the amygdala itself actually learns or tracks changes in values more rapidly than the striatum does. Okay, so that's point 1. All right, so second study. So now we're going to actually focus just on the ventral striatum. We're actually running the amygdala lesion animals on this particular task I'm going to show you now, but it's not done yet. This was done by Katie Rothenhofer, who's here and has a poster tonight. She was actually a post back in my lab and completed this study in 2 years, which is extremely quick. And now she's off doing a PhD at Pittsburgh. So now we're just going to be looking at ventral striatum lesion animals, and we're going to be comparing them to control animals. Now, here's our task. It's similar to the first task, but a bit more complex, okay? So again, it's an 80 trial block. At the beginning of the block, we're going to introduce 2 new stimuli. We're going to reverse the choice outcome mapping in the middle, and then at the end of the block, we're going to introduce 2 new stimuli. All that's the same. However, now we're going to randomly interleave 2 different types of blocks. The first block is, the first block type I call a what block, and this is basically identical to the experiment I just explained. At the beginning of the block of trials, I'm going to assign a high reward probability to one of the images, a low reward probability to the other image. The monkey has to sort that out and then pick the good image, okay? If, however, it's a where block, the animal has to learn to make a saccade in one of the directions and ignore the image that's there. So in other words, I'm going to assign a high reward probability to one of the locations on the screen and a low reward probability to the other, and it doesn't matter which image is there, okay? I'm going to randomly interleave the blocks, and I'm not going to cue the block type. So the monkey has to infer what the block type is. If it's a what block, he has to figure out what the image is. If it's a where block, he has to figure out what the direction is, okay? Reasonably complicated, but actually the animals can learn this pretty well. So again, I'm plotting here on the y-axis the fraction of times the animal chooses the best initial cue. This is my 80 trial block, control animals in black, ventral striatum lesion animals in blue, and this is the 80-20 condition from just the what block. So the ventral striatum lesion animals really struggled in this particular condition in this task. This was the worst performance we saw out of them. And we actually ran this task after having run the first experiment all the way through, and this is the same 3 ventral striatum lesion animals. So they had extensive experience on just this component of the task before we ran this, but they took a bigger hit in performance actually than the control animals did. Now, interestingly, if we look at the randomly interleaved blocks of the where condition, they're basically perfect. So they lay right on top of the control animals. In fact, if you kind of squint, they're actually slightly better than the control animals. And I'm not going to unpack all of this here, but when we introduced this task, the ventral striatum lesion animals really tried to use a where strategy, even in the what block. So they would peg away at one of the cues, even though we were rewarding the image in this task. So, conclusions from study 2. The ventral striatum does not play a role in action outcome learning. We get no deficits when they have to learn the direction of a saccade. The ventral striatum is not the critic. This is a little bit for aficionados of reinforcement learning here in the audience. Basically, there's an actor-critic architecture that people use a lot in computer science reinforcement learning, and that's been mapped into the brain to some extent, and sometimes people think that the ventral striatum is the critic. And one way you can interpret that theory is to say that the ventral striatum should maintain the values of choices independent of how those choices are executed. So that would suggest that we would see deficits in the selection of both actions and learning optimal actions and optimal images, and we don't get that. We get just specifically deficits in learning images. There's a number of other possibilities that you could use to interpret this data. One is that the ventral striatum lesion animals cannot switch from action outcome learning to stimulus outcome learning because their performance actually dropped in the what condition compared to the previous experiment. It's actually the case that they could be doing better in that condition, but since the where experiment is actually easier for them, they might just kind of, well, they do stick with it. It's actually what we see when you analyze the data, and they don't bother switching back and forth. So it could be something of a difficulty in switching back and forth. All right, so this is basically the first part of the talk is kind of a summary of things that we've done in the previous 10 years or so. And this is kind of our current take on this subcortical RL circuitry. And I'm showing the cortical inputs to this circuitry here. So this is orbital frontal cortex input and lateral prefrontal cortex input. And when I'm talking about a bit of the striatum, I really mean the whole frontal striatal circuit. So in data that I didn't show in some older experiments we did, we think the dorsal striatum is important for action outcome learning on long time scales. That's these papers. We haven't replicated basically lesions in the dorsal striatum with the what versus where test. We'd like to do that. It's also probably important to point out that our actions are eye movements, and I believe if we were using reaches, we would be in the portion of the striatum that's receiving input from motor cortex, right? Because the learning is basically over whatever representation cortex is feeding in. So dorsal striatum is doing action outcome learning. Ventral striatum is important in visual stimulus outcome learning. That's the what versus where experiment. We think the amygdala is also important for stimulus outcome learning. And I just showed you briefly a little bit of data that suggests that the striatum learns slow and the amygdala learns fast. And of course, you could wade through all the papers to see more data that's backing that up. All right, so now I'm gonna move on to just focusing on dorsal lateral prefrontal cortex. And because it's a little bit of a theme of this meeting, I think, and because a lot of us are trying to do this, I'm gonna be talking about high channel neurophysiology recordings. And we're focusing here on dorsal lateral prefrontal cortex because we had the opportunity to basically implant a lot of arrays there. So I had some older questions that I had worked on about how information scales in populations of neurons, and I wanted to try to record from hundreds of neurons if possible to address those questions. And I went into my colleague Richard Saunders' office and I asked if he could implant 3 Utah arrays. And he suggested he could do more. He said we could probably get 8 into prefrontal cortex. So I said, you know, if you can get 8 arrays in, I'm happy to record the data and try to analyze it. So he did in 2 sets of animals. He implanted 4 arrays in the left hemisphere and 4 arrays in the right hemisphere. These are these Utah arrays, which a lot of people are using. They actually work pretty good, give you pretty good units. My postdoc, Ramon Bartolo Orozco, has carried out these experiments. I'm going to show you data from 6 analyzed sessions, 3 from each monkey, where we had 585, 747, 677, 1026, 877, 500, about 750 neurons recorded simultaneously. It's a lot of spike sorting. Ramon used to smile more. He doesn't smile as much anymore. Spike sorting appears to be a lot of work. Anyway, so we're gonna answer a number of questions here. So first, so I'm gonna use the what versus where task. It's identical to what I just explained, and we're gonna look at it from the point of view of this neural activity. So the first question here is, how does the brain maintain expected reward representations across the many choices with which we are faced, right? So we're introducing new images in every block. In half of the blocks of trials in this task, the animal has to learn the values of one of those images, and somehow he probably has to keep straight or keep track in his brain the values associated with different images. Also, we reverse in the middle of the block the values of the two images, and so the question is, how do the representations change when the values of choices change? So he's going to be making eye movements when we reverse to the same images and making the same eye movements in a lot of trials, but the representation may change because the values changed. And this is really just kind of a straw hypothesis, which is, does prefrontal cortex use a common coding space to represent choices across different options? So one possibility is we get a representation that's, good object, bad object, or good direction, bad direction, and it doesn't really care about the properties of the image that's present. All right, so this is just the same behavior that I've been showing you. This is the fraction of times the monkey picks the best initial cue, trials. This is the 6 sessions from the 2 animals. This is just to show you that, in fact, these animals learned as well. This is the data from the what, and this is the data from the where condition. So we have good learning here. So how are we going to analyze this data? So it's learning data, and the first thing I'm going to do is I'm going to remove the first 4 trials. That's going to approximately get rid of this initial period back here when the monkey's sorting out what the good option is. So I'm going to remove the first 4 trials of the acquisition period. I'm going to remove the first 4 trials after the reversal period, and then I'm going to take all the trials here where the monkey picked object 1 and all the trials where he picked object 2 for the what condition, and I'm going to build a classifier which allows us to predict whether the monkey chose object 1 or object 2, right? So I'm just going to do linear classification over the 2 objects. I'm going to do the same thing with the reversal period, and I'm going to do that for every block of trials that I've recorded. So similar to what Sarah was talking about in many of the talks that I guess you've had here, I'm going to build this matrix W, where the columns are going to be these discriminant lines from each phase of each block of trials, okay, W1, W2, etc. I'm going to go up through WK for my whole recording session, and K is going to be 96. That's because I have 24 blocks of trials, 12 of each kind. I have 2 phases, and I'm actually going to use 2 time bins here, which gives me 2-dimensional decoding for each phase. Okay, so I have 96, and then I'm going to look at whether or not, right, we can approximate the WK using some low-dimensional subspace of V's, right? How much variance can we explain with a few dimensions? So this is the cumulative variance on this axis. This is the eigendimensions on this axis. To get 80% of the variance across the whole task, we need 25 dimensions. To get 90% of the variance, we need 50 dimensions. Whether that's high or low dimensional, I guess, is a matter of taste. It feels like it's fairly high dimensional to me. But more interestingly, when we break apart our W matrix into just the discriminant lines for the what condition and just the discriminant lines for the where condition, we get the following. So this is a little bit busier. So this is the unity line here. This is the cumulative variance for a random basis. The blue line is my cumulative variance as a function of eigendimensions for the what condition, and the green line is the same thing for the where condition. And the main point here is that the what condition is higher dimensional than the where condition, right? So it takes more dimensions to span the space, the task space, in the what condition than it does in the where condition. Right? So task dimensionality is higher in what blocks than it is in where blocks, which really just means that the W's are more different across what blocks than across where blocks. All right? So now I just want to unpack that result. Doing okay on time. All right, so the next thing we're going to do is we're going to take those discriminant lines W, and we're going to project the data onto those discriminant lines, and we're going to measure information in this scalar space here, right? So normally when you do classification, you project your data down and you ask whether it's above or below some threshold, and you classify that trial, but you can also measure information in the same space and information in classification performance. If things are Gaussian, are really closely related, it works pretty well in this data, which I'm not going to show you. So here's my projection. I can roll that projection over here. I'm going to measure d squared, right, which is basically our old friend d prime, which is just the squared difference between the means for object 1 and object 2 divided by the variance, right? It's just the signal to noise ratio in the data. Furthermore, I'm going to do that in a bunch of different ways, okay? So I can take my discriminant line from the acquisition phase, and I can project my data on there, and I can measure d squared. But I can also take my discriminant line from the reversal phase, and I can project my acquisition data on the reversal phase, okay? And I could take my discriminant line as well, so now I'm showing here a bunch of different discriminant lines. I can take my discriminant line from a different block of trials of the acquisition phase, and I can project block 1 data onto that discriminant line, and I can measure information in each of those different spaces. So I'm asking how much information is shared across phases of the task or across blocks of trials, okay? Shared information, right? So if these W's are orthogonal, because I'm using linear measures, then when I project my data from the acquisition phase into the reversal phase, I'll end up with 0 information. Of course, I'm never going to get 0 information because these are non-negative metrics, but that's the idea. This is the equation here, which tells me how much information I should get when I use some arbitrary classification boundary and project my data into it. I'm just projecting the means onto that, and I'm divided by the noise projected into that same subspace. All the action, actually, and I'm not going to unpack this either, is in the numerator. So it really is all about whether or not the angles between the W's for acquisition and reversal or for acquisition and other blocks of trials are related. And of course, I'm going to work my way back to the dimensionality using this approach. All right, so here's my measures. This is just from the what condition. This is when I use my W within, right? So I define my W, and this is all really heavily cross-validated, what you have to do or you get tons of overfitting. So here's W within. Here's when I use the opposite phase. So I project either the acquisition data into the reversal phase or the reversal data into the acquisition phase. I get a big drop in information. Here's another block of trials, but still the what block. And here's when I use W from the where condition and project my data onto it, all right? So I lose a lot of information when I project my data into these other dimensions, okay? So if we look at the same thing for the where condition, remember this is where the monkey's moving his eyes, right? We see that we still lose information a fair bit when we go across phase from acquisition to reversal, but when we go across blocks, we actually lose a lot less, okay? So using my decoding line from a different block of learning eye movement directions to decode this, I preserve a lot of the information, all right? So the where blocks actually control for a lot of things like just overall non-stationarity in the task, etc. All right, so here are my projections for the what condition. I lose a lot of information, which suggests that my W's are very different in the reversal phase and from different blocks of trials than they are here, and different really is the angle between them. I have a lot more shared information across blocks of the where condition, right? And so this increased orthogonality, right? This is my shared information when I use W's from different blocks of trials. The increased orthogonality here across blocks means that when I try to span my W's from all the blocks, I need more dimensions, okay? So the higher dimensionality in the what condition, the blue line here, and in the where condition is basically consistent with more shared information in the where condition and less shared information in the what condition. I can also go after that directly, so I can actually measure the angle between these manifolds, if you will, and these manifolds, right? So compare the angle between the acquisition and the reversal phase. And basically, I get something that's fully consistent with this and with this. The angle between different phases and different blocks in the what condition, when the monkey's learning objects, is larger than the angle between phases and blocks in the where condition, right? So roughly, this is preserved coding of direction across blocks of trials, which perhaps isn't that, surprising. And this is different coding for the different objects that the monkey's learning across different blocks of trials. And that different coding drives a higher dimensionality in the what blocks, across what blocks in the where blocks, and more shared information and less shared information. Okay, so that's more or less the take-home message. Right, I have a few minutes yet. I want to leave enough time for questions. So we can also kind of unpack this trial by trial as the monkey's learning, and you can do it during acquisition or during reversal. I'm just going to do it here for reversal. So what I'm plotting here is actually the neural activity projected into the acquisition space. So I define my decoding. Remember, I have 2 time bins, dimension 1 and dimension 2 here, during the acquisition phase. And I'm going to project both the acquisition trials and the reversal trials into this space. The red circle and the blue circle are kind of the mean and, I can't remember, I think 1 or 2 standard deviations, maybe it's 2 standard deviations, of neural activity for selecting option 2 during the acquisition phase, and this is for selecting option 1. The orange dots are the reversal data for option 1. I'm sorry, Yes, for target 2. And the blue dots are target 1 reversal data, okay? So I already said, basically, that there's more separation, meaning more information, when I use my within classification boundaries to project the data, and there's less when I project the reversal data in. So we can just see that here, right? The dots collapse. So the orange and cyan dots are closer together than the red and the blue dots, and that's just exactly that. Now, if we project into the reversal space, I'm projecting basically the acquisition and reversal data in the reversal space, and I'm keeping the colors. Now we see that the data is moved far apart for the reversal data, but it collapses, right, for the acquisition data. This is the same data, just projected into 2 different spaces. And what you can see if you follow the orange dots here, numbered 1, 2, and 3, this is the first trial after reversal, the second trial after reversal, and the third trial after reversal, right? We don't tell the monkey when we reverse the choice outcome mappings. So the first time the monkey selects that other image, he's doing it under the assumption that that's still the best image and nothing has changed, right? But maybe he doesn't get rewarded there, he selects it again and he doesn't get rewarded, and he starts to get the idea that he's not being rewarded, and at that point in time our representation changes, okay? I've also got the 1, 2, and 3. Here's 1, 2, and 3 is stacked up here. So those are the first 3 trials after the reversal for this other target, and then eventually I kind of converge to this stationary distribution. So we can measure information about the acquisition data in the acquisition and reversal spaces as a function of trial relative to reversal, right? So trial 0 here is an alignment of where I reversed the choice outcome mapping in each block of trials. I've aligned the data. This is the distance basically between single trials and that stationary distribution in the acquisition space. So it starts out high in the acquisition space. This is actually a difference in information. That's why it can go negative. We have a large distance in the acquisition space, but then when we reverse the choice outcome mappings, we transition, and then we end up with a very small, actually an even negative distance. For the reversal data, we start out with no distance between the single trials in the reversal space, and then when he learns that the other space is relevant, we transition to having a high distance. So basically all those bar plots I've been showing you is a measure of out here, and this is showing how he transitions with learning, and we see something similar in the where condition. I'll just point out here quickly that there's some preserved information in the where condition relative to the what condition. This is what I was showing you in the bar plots, and that's why this is actually above 0 and why this one actually drops. So we could do the same trick for acquisition, but it's the same thing basically. All right, so conclusions for this part. How does the brain maintain expected reward representations across the many choices with which we are faced? It basically drives neural activity, at least for the what condition, when you're learning across different objects, into new parts of space, okay? Interestingly, when we reverse the choice outcome mapping, so the value associated with each of the choices, we also drive activity into a new part of space, even though it's actually at that point in time the same actions and the same objects. All we've done is reversed the reward mappings to the actions. Actually, it's to the objects, to the actions. We end up with more preserved information. So prefrontal cortex does not use a generic coding space to represent choices, at least choices over objects. And then because learning values over a new pair of objects or reversing the values drives activity in a new part of coding spaces, this increases the accumulated dimensionality of dIPFC representations, right? So when we look at this accumulated dimensionality over the whole recording session, it's higher for the what condition because we've driven activity into different parts of space when the monkey learns the values of those parts of space. All right, so just to summarize, I'll come back to my diagrams here. So in the first part of the talk, I just showed you that the amygdala actually plays an important role in reinforcement learning. The striatum also plays a pretty important role, but it's fairly specific in the ventral striatum to learning stimulus outcome mappings. We always use visual stimuli. And then I was talking about a lot of data here from dIPFC. I don't know if we would see pretty similar representations in the caudate if I did physiology there and could do a lot of record from a lot of channels. My guess would be I would see something pretty similar. And because I'm at Cosyne and we have a lot of modelers here, I just want to throw this out. A feed-forward kind of model actually works reasonably well when you're thinking about visual cortex, right? So deep networks give us kind of a decent metaphor for what's going on there. But we have an extremely looped architecture in prefrontal cortex. There are many routes to get through this system to get information into any of the nodes. And there are just recurrent connections everywhere here, loops, et cetera, et cetera. And I think actually we need to push into developing good models for complex circuitry like this in the front of the brain, And there are a few things around, but there's not a lot. And as someone who primarily runs an experimental lab and looks at the theory literature for ideas about how to think about my data, I would encourage people here who do this kind of work to begin thinking about more complex circuitry like this that have many nodes interacting. That's just a quick picture of the lab. I showed you all the individuals who were doing each of the pieces of work. The only last thing I want to mention, I mentioned Richard Saunders, Phil Browning assisted with that surgery. The lesion animals actually came from my collaborator, Betsy Murray. These are the people I pointed out on the various slides. And all my funding comes from the intramural research program. Thank you.
Thank you very much for this very interesting talk. It wasn't clear to me the where task is much easier. It's just binary, right? Always right is a reward and left there's none, and then it switches as well, like in the what task?
In each block of trials, he has to learn whether it's left or right at the beginning of the block.
Yeah.
But it is true, it's always left or right, whereas the images differ. But if you look at the behavioral performance, most of the time it's pretty similar across the two. So it's not like they can actually learn more quickly in the where task.
But it's only one bit versus like 20 images or so.
Well, in each block, though, it's actually only 2 different images.
Only 2, okay.
Yes, yeah.
Okay, questions? Maybe over there?
Okay, yeah, thank you for a great summary of your long-time work. So regarding the comparison between the amygdala and the ventral striatum, so now the amygdala is known to have different components. As probably you know, the lateral and basolateral amygdala is closer to the cortex, and the central amygdala is closer to the basal ganglia. So if you specifically lesion the central amygdala, what will be the difference between the ventral striatum?
That's a good question. I would actually guess that it would be lesions to the basolateral, so the collection of nuclei that are under, that would give you results more similar to the ventral striatum lesions, mostly because they're actually projecting to the ventral striatum. But we haven't really tried that, so I'm not sure.
Hi, so my question is, what type of information is being measured as independent across the different blocks, or so I just don't know, what was the information that was being measured, or
So the information that we measured was just specific to the condition that was relevant in that block. So in the what blocks, the information was whether or not the animal chose object 1 or object 2, and in the where blocks, it was whether the animal made a saccade left or right. Does that make sense?
Yes, so it was just 2 types of information.
It's different in the 2 blocks, yes.
There's one question over here.
Hey, I was wondering, because you said there was like the amygdala is learning quickly and the striatum is learning slowly. Do you think there's any advantage in terms of learning to having these systems that learn at different speeds? Maybe because also your task has different timescales between that of trials and that of blocks. Like, do you think they interact in a way to help learning?
Yeah, I mean, basically, if you think that they're really just kind of doing classical slow value update kind of learning, then having different learning rates makes you more optimal for different kinds of environments. So the amygdala, because we tend to look at flexible learning where we change the stimulus outcome mappings over time, the amygdala is going to be a little bit better at tracking rapid changes in value. The ventral striatum is going to be better at tracking slow changes in value. The issue with the amygdala is that if there's noise in the reward outcomes that has to be kind of filtered out to get an accurate estimate of the value, the amygdala is going to be more sensitive to the noise if the noise tends to be independent noise across trials. So the amygdala is going to be more likely to track the noise with its high learning rate, whereas the ventral striatum will be a little bit better at integrating over the noise.
If we have time for one more, I just wanted to ask about if you would speak more about exploration during this task and what you see.
Yeah.
In either ventral or
yeah.
LPFC.
That's a great question. So we have another experiment that we have been using to study the explore-exploit trade-off. Because in these bandit tasks, you really, particularly the easier schedules, the exploration's kind of over in a couple of trials, like 1 or 2 trials. It happens pretty fast. We have another experiment where it's a 3-arm bandit, and we periodically replace one of the stimuli with a different stimulus, and then we can look at whether the monkey checks out that new stimulus. There, basically, we get, and you can calculate an exploration bonus for the animal, how much it should be worth to check out that stimulus. That exploration bonus is pretty strongly encoded in both the amygdala and ventral striatum, but I think it's more strongly encoded in the amygdala, but I'm going by memory, so I'm not sure.
Maybe while Tom sets up, I have another question. We heard now all these, a few talks about dimensionality of the neural code, and I would like it seemed like you needed many principal components to get up to the high variance, but the number of stimuli, so as to say, in your task was pretty small, right? So how do you see your data in this context of the talk we heard from Ken Harris and maybe Sarah Sawyer from before?
Yeah, I mean, I think to some extent it's kind of a related mechanism, because every time I introduce a new pair of stimuli, I get a little bump in the number of dimensions. If you look, though, that dimensionality expansion in prefrontal cortex doesn't appear in the first few trials. It only appears after the monkey learns the values of the 2 options, and then you actually get the expansion. In the early trials, when the animal's exploring around, it's actually a much more compact space. I didn't actually show that, but so I think it's a related mechanism, but it also depends on the learning of the animal.
But there's an agreement, you would say, with
yeah. Although I haven't looked at what happens if you scale this to really, really large numbers of images.
Okay, let's thank Bruno one more time.